So, the stages of DNA replication begin when the DNA unwinds. So the first stage of DNA replication begins with the unwinding of the double helix. And that double helix is going to unwind by breaking the hydrogen bonds between the nitrogen bases. And those hydrogen bonds aren't just going to break on their own, they need an enzyme to break them, and the enzyme that breaks them is called helicase. The second part of DNA replication happens when the free nucleotides that are floating around in the nucleus, sort of like this, we have some free nucleotides here that are just sort of floating around. They're going to start to bind to places where there is a nitrogen base exposed. So those free nucleotides are going to start to look for a place to bind. So in your notes it says the free nucleotides form hydrogen bonds with their complementary base pairs. And the complementary base pairs just mean that adenine is going to bind with thymine and guanine is going to pair with cytosine, just like we've talked about before with DNA. A goes with T and C goes with G. And these pairs aren't going to form on their own, just like before we need a helicase to break the bonds. Now we need another enzyme to be able to make that bond form again. And the enzyme that we need is called polymerase. So polymerase is the enzyme that's going to bind the free nucleotides to the template strand. The last thing that's going to happen in DNA replication is that the new DNA strand will rewind, and it's going to coil itself up to form a double helix. And the result of DNA replication is that you get two new strands that are identical to the original strand. They're exactly the same. So that is DNA replication. Now that you have all of your blanks filled in, we're going to go ahead and label the parts of the picture over here. And we're going to label each of the bold words in your notes will be a label on the picture. So let's start up at the top. Your first bold word is double helix. So in your picture, our double helix is this twisted part of the DNA strand. So this whole part here where it's twisted, that shape is called a double helix. So we're going to label it. The next blank that you have on your notes is hydrogen bonds. Your hydrogen bonds are going to be right between the nitrogen bases. So right between the C and the G, or the A and the T, all the way down the strand of DNA, between each of those nitrogen bases, there is a hydrogen bond. So we're going to label one of them. Right here we have a hydrogen bond. Alright, if we go back to your notes, your next bold word was free nucleotides, and your free nucleotides are going to be found down here, floating around in the nucleus. So we're going to label all of these up here. Those are the free nucleotides. And then in your notes it wants you to label adenine, thymine, cytosine, and guanine. So anywhere on this picture where you see an A, that would be adenine. Anywhere you see a T, that would be thymine. So we're going to go ahead and label any one of those. This will be adenine. This will be thymine. And then we can label guanine right here. And we have a cytosine right down here. So we have our 4 nitrogen bases labeled. Anywhere you see those letters, it's the same nitrogen bases. And the last, we missed replication fork. So let's go ahead and label replication fork in the picture. That is where the DNA starts to unwind. So right about here where you get a division in the strands of DNA, that is the replication fork. And then the last thing you need to label in your notes are the new strands of DNA. The new strands of DNA are going to be down here at the bottom of the picture. We have one on this side, and also one on this side. So this is going to be the new DNA. And there we have your picture all labeled. Okay, we're going to talk about DNA replication. Before we get into DNA replication, I want to remind you of when we have talked about DNA replication before. And we already talked about it when we talked about the cell cycle. So if you remember the cell cycle wheel, it has 4 parts. Those 4 parts are called G1, S, G2, and M. And we studied what each of these parts included. The M phase is mitosis. It has the 4 stages of mitosis and cytokinesis. So it has prophase, metaphase, anaphase, telophase, and cytokinesis. G1 is where the cell grows in size. It increases in mass. G2 is where the cell prepares for mitosis. And the S phase, which is what we're going to be talking about today, is where DNA replication happens. So the DNA is going to copy itself. All right,